Hello students, now let us start with the new concept of the molecular basis of inheritance, that is packaging of DNA helix or the nucleosome model. Okay, so what is the size of the cell? What is the size of the cell? That is in terms of 10 to the power minus 6, that is in the micrometer. Okay, what is the size of the nucleus? So it is also in the micrometer. What is the size of one DNA? So now kaltidivi, that is the size of that is the distance between the each conjugative base pairs in a DNA is 0.34 into 10 to the power minus 9 meter, andre 0.34 nanometer, or in other words 3.4 angstrom. That is distance between each conjugative base pairs. And the number of base pairs in the double helix structure of DNA 6.6 into 10 to the power plus 9 base pairs. Distance between each conjugative base pair is this much, and there are about 6.6 into 10 to the power 9 base pairs are present. Erdanna multiply madbitri antandre you will get the length of the DNA, that is 2.2 meter. Length of the DNA is 2.2 meter, whereas the size of the nucleus is 10 to the power minus 6 meter. So this long DNA should be packed inside the small nucleus. Okay, that is by the nucleosome model ಅಥವಾ packaging of the DNA helix ಅಂತ ಕರಿತೀವಿ. Okay, so ಬಹಳ ಸಿಂಪಲ್ ಆಗಿ ಅದ ನೋಡೋಣ, ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳೋಣ. Inside the nucleus there are some specialized proteins, basic proteins. Those are called as the histone proteins. So, inside the nucleus there are some specialized specialized basic proteins are present. Those are called as the histone proteins. Okay, histone proteins anta karitiri. There are 5 types of histone proteins, that is H1, H2A, H2B, H3 and H4. These are the positively charged specialized basic proteins called histone proteins. 5 histone proteins are present. Okay, these 5 histone proteins are positively charged. Yake positively charged? Because they having the basic amino acids called lysine and arginine. These carries the positive charges, hence these are the positive charges. So out of these 5, these 4 histone proteins are arranged in the 8 octo fashion. Okay, and hence these forms the octomer, histone octomer anta karitivi. Iga H2A, H2B, H3, H4 idr hindenu kuda same irtada. Okay, this forms the histone octomer or new body anta karitiri. ಅರ್ಥ ಆಯ್ತಾ? ನ್ಯೂ ಬಾಡಿ ಅಂತ ಕರಿತೀರಿ. ಇವಾಗ ಬರೋಣ ನ್ಯೂಕ್ಲಿಯೋಸೋಮ್. ಹಂಗಂದ್ರೇನು? Nucleosome means the process of wrapping of negatively charged DNA over the positively charged histone octomer is called as the nucleosome. What is the nucleosome? The process of wrapping of the negatively charged DNA over the positively charged histone octomer ಅಥವಾ histone proteins is called as the nucleosome. Okay, nucleosome ಅಂತ ಕರಿತೀರಿ. ಇಲ್ಲಿ ಬಂತು ಹಿಸ್ಟೋನ್ ಆಕ್ಟೋಮರ್. ಹಂಗಿದ್ರೆ ಹಿಸ್ಟೋನ್ ಅಂದ್ರೆ ಏನು, ಹಿಸ್ಟೋನ್ ಪ್ರೋಟೀನ್ ಅಂದ್ರೆ ಏನು? ಸೊ ಅದನ್ನೇ ನಾನು ಇಲ್ಲಿ ಎಕ್ಸ್ಪ್ಲೈನ್ ಮಾಡಿದ್ದು. ವಾಟ್ ಇಸ್ ದ ಹಿಸ್ಟೋನ್ ಪ್ರೋಟೀನ್ಸ್? The positively charged basic proteins are called as the histone proteins. There are 5 histone proteins: H1, H2A, H2B, H3 and H4. Okay, out these histone proteins containing some basic amino acids like lysine and arginine, because of which they are positively charged. ತಿಳಿತು? And out of this, these 4 proteins arranged in the octamer fashion, and hence it forms the histone octamer or new body over which the negatively charged DNA is wrapped. Okay, that forms the nucleosome. ತಿಳಿತು? ಸೊ ಈ ರೀತಿ ಅರೇಂಜ್ಮೆಂಟ್ ಆಗಿರ್ತದ. ಅಂಡ್ ಒನ್ ಮೋರ್ ಇಂಪಾರ್ಟೆಂಟ್ ಥಿಂಗ್ ಮುಂದೆ. So this is histone octamer. On this histone octamer, negatively charged DNA illodu wrapping aitu. Then a one protein, that is called as the H1 protein, non-histone protein anta karitivi. That protein separate such nucleosomes. ಈ ನ್ಯೂಕ್ಲಿಯೋಸೋಮ್ಗಳನ್ನು ಸಪರೇಟ್ ಮಾಡ್ತದ H1 ಪ್ರೋಟೀನ್. ಇದೊಂದು ಬರ್ತದೆ. ಮತ್ತು ಇದಕ್ಕೆ ರ್ಯಾಪಿಂಗ್ ಆಗಿ ಅಗೈನ್ ದೇರ್ ಯು ಫೈಂಡ್ H2A, H2B, H3, H4. So again wrapping continues. So again H1. So like this it will continue. So like this it will continue. So this H1 protein is also be called as the plugging protein ಅಂತೀರಿ, or it is also be called as the marker protein ಅಂತನೂ ಕರಿತೀವಿ. So like this repeated units are formed. Okay, so in the nucleus there are about 200 base pairs. Okay, irtada. And inside the nucleus such repeated units, okay, such repeated units, that is strained bodies are formed. Those are called as the chromatins. What are the chromatins? The repeated structures athwa repeated units of the strained bodies found in the found in the nucleus are called as the chromatins. What are the chromatins? The repeated units are the repeated strained bodies athwa colored bodies found in the nucleus are called as the chromatins. Okay, these DNA helix forms the repeated units of the strained or colored bodies in the nucleus. Those are called as the chromatins anta karitiri. Okay, so these chromatins further forms the chromatin fibers. ಈ ಕ್ರೊಮ್ಯಾಟಿನ್ಗಳು ಮುಂದೆ ಏನಾಗ್ತಾವು? ಕ್ರೊಮ್ಯಾಟಿನ್ ಫೈಬರ್ಸ್ಗಳು ಫಾರ್ಮ್ ಆಗ್ತಾವು. ದೀಸ್ ಕ್ರೊಮ್ಯಾಟಿನ್ ಫೈಬರ್ಸ್ undergoes condensation during the mitosis of the cell division. Okay, these chromatin fibers undergoes a condensation during the mitosis of the cell division. ಅದರಲ್ಲಿ ಮೈಟಾಸಿಸ್ ಅಲ್ಲಿ ಯಾವುದದು? ದಟ್ ಈಸ್ ದ ಮೆಟಾಫೇಸ್ ಸ್ಟೇಜ್ ಅಲ್ಲಿ, ಇನ್ ದ ಮೆಟಾಫೇಸ್ ಆಫ್ ದಿ ಸೆಲ್ ಡಿವಿಷನ್, and hence they forms the chromosomes. Okay, ಇಲ್ಲಿ ಬಹಳ ಗಮನ ಕೊಡೋದೇನು, inside the nucleus there are different about 200 base pairs of the DNA helix are formed. These DNA helix, the repeated units of the DNA helix forms the strained bodies or colored bodies. Those are called as the chromatins anta karithivi. Okay, these chromatins are looks like nodo beds on string structure when viewed under the electron microscope. These beds on string structures, they forms the chromatin fibers. These chromatin fibers undergoes condensation, okay, in the metaphase stage of the cell division and forms the chromosome. Okay, chromosome anna form ಮಾಡ್ತದ. ಅರ್ಥ ಆಯಿತು. ಸೊ ಇಲ್ಲಿ ಕ್ರೊಮ್ಯಾಟಿನ್ಸಲ್ಲಿ ಮತ್ತೆ ಎರಡು ಟೈಪ್ ಮಾಡ್ತೀವಿ. ಯಾವ್ಯಾವು? ಒಂದು EU ಕ್ರೊಮ್ಯಾಟಿನ್, ಅನದರ್ ಒನ್ ಈಸ್ ದ ಹೆಟರೋಕ್ರೊಮ್ಯಾಟಿನ್. ಬಹಳ ಇಂಪಾರ್ಟೆಂಟ್. ಒಂದು EU ಕ್ರೊಮ್ಯಾಟಿನ್, ಇನ್ನೊಂದು ಹೆಟರೋಕ್ರೊಮ್ಯಾಟಿನ್. What is meant by the EU chromatin and heterochromatin? EU chromatin means the highly strained, EU chromatin means the loosely arranged and less stained. Loosely arranged, less stained chromatins are called as the euchromatins, and these euchromatins are more active compared to the heterochromatins. And heterochromatin means highly stained ಅಥವಾ dark stained and tightly packed. ಇದು ಲೂಸ್ಲಿ ಪ್ಯಾಕ್ಡ್, ಇದು ಟೈಟ್ಲಿ ಪ್ಯಾಕ್ಡ್. and dark stained chromatins are called as the heterochromatins, and these heterochromatins are comparatively they are less active ಅಥವಾ inactive ಅಂತ ಹೇಳ್ತೇವೆ. ತಿಳಿತು? ಸೊ ದಿಸ್ ಈಸ್ ಅಬೌಟ್ ದ ಪ್ಯಾಕಿಂಗ್ ಆಫ್ DNA ಹೆಲಿಕ್ಸ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಓಕೆ, ಸೊ